Good morning. It's May 19th, and this is your daily brief in science. Here's everything you need to know. A recent study presented at the ATS 2025 International Conference showcases a promising advancement in the management of obstructive sleep apnea, or OSA. Researchers highlighted a wearable pulse oximeter paired with a connected software platform designed to monitor treatment responses effectively. This technology aims to improve patient access to sleep apnea care, allowing pulmonologists to manage treatments more efficiently without frequent referrals to sleep specialists. Unlike consumer devices such as the Oura Ring and Apple Watch, this pulse oximeter utilizes transmittance-based photoplethysmography. This method provides superior signal quality and enables continuous data collection, offering significant advantages over traditional in-lab polysomnography. The device allows for ongoing monitoring, which helps clinicians observe night-to-night variability in treatment responses. Clinical trials indicate that the device is user-friendly, with high utilization rates among patients. Notably, 85% of participants exceeded the recommended usage of 9 nights over 47 days, with some wearing the device for as long as 44 consecutive nights. Patients can also access their health data through a smartphone app, fostering better communication with healthcare providers. While the pulse oximeter has received FDA clearance, the connected software still requires further validation for broader clinical use. Researchers are actively working to gather more data to enhance understanding of patient and clinician experiences with this device. This medically approved technology prioritizes user comfort and continuous tracking, marking a significant step forward in the management of OSA. A new multiplex surrogate virus neutralization test, or SVNT, developed in Singapore, is making waves in the global health landscape. This innovative test is now utilized in over 90 countries to detect antibodies against a range of viruses, including variants of SARS-CoV-2, Ebola, and Henipaviruses. Led by researchers Prof. Wang Linfa from Duke NUS Medical School and Dr. Tan Chi Hua from NUS Medicine, the SVNT not only assesses immunity levels, but also plays a crucial role in evaluating vaccine efficacy. One of the standout features of this test is its safety profile. It does not require live viruses for testing, instead using a harmless spike protein from the virus, which minimizes infection risks. The SVNT also enhances contact tracing during epidemics by identifying previously infected individuals, thereby revealing transmission chains. In response to the COVID-19 pandemic, Singapore has taken significant steps to bolster its pandemic preparedness, launching a $100 million program for research in epidemic preparedness and response in 2022, alongside the establishment of the Communicable Diseases Agency in April 2025. This initiative reflects a growing awareness of Disease X, a term that signifies unknown pathogens that could lead to future global pandemics, highlighting the necessity for improved preparedness. Researchers are now looking to develop a more comprehensive test capable of detecting all pathogens on the World Health Organization's priority list for antibiotic-resistant bacteria. The SVNT evolved from the CPAS test, which was the first commercial tool to detect neutralizing antibodies for SARS-CoV-2, addressing limitations posed by evolving variants. Its international adoption is evident, with institutions like the University of Oxford and the University of Melbourne employing the test for infectious disease research and surveillance. High levels of neutralizing antibodies detected by the SVNT suggest likely protection against future infections, aiding in the evaluation and formulation of vaccines. The development of this multiplex SVNT was supported by the Integrated Innovations in Infectious Diseases grant, funded by Singapore's National Medical Research Council. Furthermore, the test's versatility is underscored by its application in animal studies, such as tracking SARS-CoV-2 transmission to deer in the U.S. Lift-Biosciences has unveiled a new class of immunotherapy known as immunomodulatory alpha neutrophils, or IMANS, which aims to combat solid tumors through a unique combination of direct cytotoxicity and immunomodulatory effects. This groundbreaking development was showcased during the ISCT 2025 annual meeting, held in New Orleans from May 7th to May 10th, 2025. Mark A. Exley, the chief scientific officer of Lift-Biosciences, presented findings in a session titled IMANS, a transformative solution for overcoming challenges of solid tumor immunotherapy and rejuvenating innate immunity. The presentation highlighted the potential of IMANS to address the limitations faced by existing cancer therapies. Preclinical data indicated that IMANS can infiltrate tumors for over 21 days, effectively recruiting natural killer, or NK cells, which suggests a capacity for long-lasting immunity against cancer. In both in vitro and in vivo studies, IMANS were found to convert cold tumors into hot tumors, thereby enhancing systemic anti-cancer immunity through cytokine secretion and increased activation of CD8-positive T cells. The studies demonstrated that IMANS effectively penetrate various solid tumors, including non-small cell lung cancer, bladder, pancreatic, and colorectal cancers, exhibiting strong anti-cancer properties. CEO Alex Blythe emphasized the importance of a scalable, GMP-compliant manufacturing process for producing high-quality IMANS, which could lead to more effective treatment options for resistant tumors. Lift-Biosciences is actively developing a portfolio of engineered IMAN therapies targeting a range of solid tumors, leveraging their patented N-Lift platform. Unlike traditional therapies that primarily focus on adaptive immunity, IMANS are designed to address the immunosuppressive environments of tumors, offering a more comprehensive treatment approach. Additional research has shown that IMANS lead to significant increases in CD8-positive T-cell activation markers and sustained NK cell survival in co-culture experiments. Researchers from the University of Utah Health, University of Washington, and PacBio have made a significant breakthrough in understanding human DNA by mapping regions that mutate rapidly. This study reveals that our genetic material may evolve much faster than previously thought. Unlike earlier research focused on stable parts of the genome, this team has uncovered highly mutable regions that change nearly every generation. Utilizing advanced sequencing technologies, the researchers achieved high-resolution images of genetic variation, allowing for a thorough analysis of both small and large DNA changes. Their findings suggest that each human has approximately 200 new genetic changes compared to their parents, with many of these occurring in the previously hard-to-study regions of DNA. This study was published in the journal Nature on April 23, 2025. The implications of this research are significant for genetic counseling, as it can clarify whether diseases in children are inherited from parents or arise from new mutations. Understanding these mutation patterns impacts predictions for future disease risks for siblings. Additionally, the sequencing data from this study will be publicly available, promoting further exploration into human evolution and genetic diseases. The research utilized a unique multi-generational Utah family that has contributed DNA samples since the 1980s, offering a valuable resource for tracking genetic variation over time. Future studies aim to apply these sequencing techniques to additional families to explore variations in mutation rates, which could further refine predictions for disease risk and genome evolution. Lynn Jordy, Ph.D., emphasizes that mutation rates are as fundamental to human biology as the speed of light is to physics, highlighting their role in distinguishing humans from other species. A remarkable case in Colombia is shedding light on the complexities of Alzheimer's disease. Aliria Rosa Piedrahita de Villegas, a resident of Yarumal, carried the E280A mutation linked to early-onset Alzheimer's, but did not develop the disease, despite her family's history of the condition. Researchers, including Dr. Kenneth Kosick and Dr. Francisco Lopera, conducted a detailed study of her brain. They discovered that alongside the Alzheimer's gene, she had a rare protective mutation. This unique genetic profile included 2 copies of the apolipoprotein, or APOE mutation, which may have played a key role in her resistance to the disease. Interestingly, Piedrahita de Villegas' brain showed high levels of amyloid plaques, a hallmark of Alzheimer's, but very few tau proteins. This finding suggests a potential protective mechanism against the disease. The APOE mutation produces lipoproteins that may counteract the harmful effects of amyloid plaques, offering valuable insights into future Alzheimer's treatments. Yarumal has a notably high prevalence of Alzheimer's due to the E280A mutation, impacting nearly 6,000 residents. This mutation is inherited from a common ancestor and often leads to symptoms appearing in individuals as young as their 30s. With the global Alzheimer's population projected to reach 115 million by the year 2050, understanding cases like Piedrahita de Villegas's could pave the way for breakthroughs in prevention and therapy. Dr. Kosick highlighted the importance of her unique genetic profile for future Alzheimer's research and treatment development. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.